Alright guys, welcome. Before you begin the lesson, please take a moment to write down these 3 questions and answer them. So hit pause, get these questions written down, take a guess at your answer before you continue playing with the video. The purpose of this video is for you guys to learn how materials can be transported into and out of cells. And there's 2 main categories, and the first category is called passive transport. Key things you need to know about passive transport is passive transport does not require energy. The second thing you need to know is kind of like this slide. During passive transport, materials are moved from an area of high concentration to an area of low concentration. So just like when you are going down a slide, you sit at the top, you end up at the bottom, you go from high to low, and it doesn't take any effort on your part, similar to the cell not needing to expend any energy to make this happen. Now, you may be thinking to yourself, I understand the whole slide thing, you slide down a slide, that's all cool, but what was that concentration word she was saying? In science terms, the concentration is the amount of solute compared to the amount of solvent. You're like, awesome, more words, I still don't understand. That's okay. In normal people terms, this is usually the amount of good stuff compared to the amount of water that it's dissolved in. So the easiest way to explain this is with Kool-Aid. You dissolve the sugar and the food coloring in the packet of Kool-Aid into water. The more Kool-Aid, or good stuff, you have compared to the amount of water, the more highly concentrated it's going to be. You're going to have a stronger flavor, darker color. Now, if you have one of those moms that thinks that Kool-Aid is way too sugary, and they always mix it using twice as much water as they should, then you're going to get something with a lower concentration, sometimes said to be diluted. You have more water or solvent than you have good stuff, also known as solute, whatever is dissolved into the water. Now, there are 3 main types of passive transport. The first one is diffusion. Diffusion is simply the movement of molecules. So these dots are going to represent my molecules from an area of high concentration to, over time, an area of lower concentration. This can happen inside cells, outside cells, and through the cell membrane. A great example of this is spraying perfume. You spray it at one end of the classroom or in your bedroom, and within a few minutes, the perfume molecules have spread out and you can smell it throughout your entire bedroom. Or worse, if the kid in class sitting a few seats down has gas, he may fart, and within a few moments, those molecules will spread out, and you and all the unfortunate people sitting next to you will also smell those lovely molecules. The second one is facilitated diffusion. This means that the cell needs help in order to have those particles moved. So maybe the particles are too large to be able to get through that cell membrane. They try, but they just get blocked. So instead, there are special proteins called transport proteins that act as channels. They're hollow, so those larger molecules would be able to move through those protein channels in order to go from an area of high concentration of whatever molecule we're talking about to an area of lower concentration inside the cell. And last is osmosis. Osmosis is a special type of diffusion, and it's the one that you're going to be spending the most time in class learning about. Osmosis is the diffusion. It's really hard to write on these things. Of water across, wow, this looks horrible. I apologize. A cell membrane. So we're not just talking about diffusion of anything, it's only the diffusion of water. And one of the really cool and important things that you need to understand is see how tightly packed all of these phospholipid molecules that make up the cell membrane are? Well, unless something gets to use one of those protein channels, most molecules and compounds are way too large to get through that membrane. That selectively permeable membrane will not select to let them through. However, water is one of those special things that is allowed to pass through the membrane. Alright, so let's do a little demonstration. So these 3 circles represent cells, and they've been placed into 3 different glasses of salt water, something of the sort. To make things simple, since this may be the first time you've been introduced to osmosis, we're just going to represent the water. So all these blue dots represent molecules of water. So in this first case, over here on the left, right now, the distribution of water molecules is about even on the outside as it is on the inside. But if something were to happen to cause the concentration of water outside that cell to become much greater than the concentration of water inside the cell. So maybe inside the cell has water mixed with salts and sugars. When this happens, the cell and those molecules naturally want to even things out. So as a result, those water molecules are going to be moving into the cell, and the cell may increase in size or even burst. Now, in our next example here in the middle, the concentration of water outside and inside the cell is going to remain fairly stable. In this situation, the amount of water moving in and out of the cell is going to be about the same. Water will always be moving, but it's going to move in at the same rate that it moves out. And as a result, there's going to be no change in concentration and no change in cell size. And last, sometimes you have the crazy example of there would be far more water inside the cell than in the area surrounding the cell. This happens if you were to dunk the cell into really, really salty water, something where the environment around it has a lot of other good stuff. Remember, that solvent dissolved in it. And in this case, in order to even things out, water is going to primarily leave the cell, and the cell could shrivel up and lose the ability to function. Alright, now we're going to add in the concentration of the good stuff, or the solute, whatever was dissolved in the water. So if we go back to this first one, I said that the concentration of water on the outside is really high, and the concentration of water on the inside was originally really low. Well, this would be because of whatever else was mixed in the water. So let's say we're dealing with salt. These red dots are going to represent the salt. That means that there is a lot of salt inside the cell. It's taken up the space where those water molecules used to be. And there'd be very little salt concentration on the outside of that cell. Now we have a special term to describe this. When the concentration of the dissolved good stuff is very low outside of the cell, we say that it is hypotonic. Hypo meaning below or low, like a hippo sinks low. So we have a higher concentration of dissolved salt inside the cell than we do outside the cell. Now in the second case, the concentration of dissolved salt inside and outside the cell is about even. We call that isotonic. Iso meaning same, and tonic referring to the particles and stuff that are dissolved in the water. And then last, if the concentration of water outside was low, that was because of all of the dissolved salt or solute. So in this case, we have taken that cell and dunked it in a cup of very salty water. So the concentration of salt outside the cell is much greater. We call this a hypertonic solution. Hyper meaning above or more. There is more stuff outside the cell than there is inside the cell. Alright, so overall, the reason I showed you just the water first is because it's only the water that's moving. But then I had to add in the other stuff so you can understand these three terms that you are going to be required to understand. So once again, a review. If it's hypotonic, there is more dissolved things inside the cell, meaning the concentration of water is going to be lower, and the water is going to move inside the cell. Isotonic, the concentrations are the same both inside and outside the cell, so the water moves evenly. And if it's hypertonic, all of the dissolved good stuff is outside of the cell, and the water is going to try to move outside in order to even out the concentrations. Alright, now I said at the beginning that there were two main categories of transport, the way the cell can move things in and out, and I'm just going to briefly introduce the second one, which is active transport. The main difference you need to know is before we moved from high to low. We went down that slide like good rule-following people. Well, active transport's a little bit more fun. This is that kid. Come on, drawing tool. This is the kid that refuses to follow the directions and play nice and chooses instead to climb up the slide. So in active transport, you're moving from low concentration to a higher concentration. And if you haven't done this in a while, you may want to try. Climbing up the slide does require energy. It requires a little more effort than sitting at the top and sliding down. Okay, once again, I have some questions for you to answer at the end. So please pause the video, copy down, and answer your questions. Complete sentences.